Contraction-Stimulated. In the past decade or so, these two topics have begun to intersect more and more, not only in the literature. For some reason, I can't advance my slide. All right, there we go. Not only in the literature, as you can see, I've just labeled three of the several reviews that are out right now looking at the effects of time of day on exercise, not only on exercise performance, but on the beneficial effects of exercise when it's done at one time of day versus done at another time of day. And it's not only in the literature and in academia, this is broadly being investigated in popular culture as well. So here's an article from the New York Times, the best time of day to exercise for metabolic health. And this is actually a recent article from BBC that just came out on January 6th. What's the best time of day to exercise? So this is something that a lot of people, both in popular culture and people who are not in academia or science, are talking about and thinking about kind of in the health and wellness sphere, but also that people are investigating intensely as well in academics. And so I think we all know that diabetes is an issue and that diabetes is a rising issue in this country, and that the main detrimental effects of the disease are due to high glucose concentrations in the blood. And these, in the instance of type 2 diabetes and metabolic syndrome, and basically every syndrome involving impaired glycemic control is on the rise. And exercise is a really great way to reduce risk of these diseases. So, for example, this is a figure from out of the Diabetes Prevention Project, where they looked at a large cohort of people who had prediabetes and looked at three different variables, whether they received basically no intervention, metformin, which is a drug commonly prescribed to treat diabetes, or they had no drug but had a lifestyle intervention that consisted of, broadly speaking, healthier eating habits and exercise, specifically aiming for 150 minutes or more of moderate intensity exercise per week. And as you can see from this figure, the lifestyle intervention had a greater effect than even the drug itself, metformin. So exercise is a great low cost, it can be free even way to reduce risk of developing diabetes. And so just to kind of outline what I'm going to be talking about today, I'm going to be talking about three main studies. The first study is going to look at the role of time of day regulation on intrinsic muscle contractile properties and contraction-stimulated glucose uptake. The next study I'll be talking about looks at time of day regulation of mitochondrial function in muscle. And then in the third study, we're going to go a little bit more mechanistic and look at the specific role of two proteins called P300 and CBP and see what their role is in contraction-stimulated glucose uptake. So the whole field of chronobiology or circadian biology, those are a couple of things people call it, but the whole field really started when people started describing this phenomenon where athletes seem to perform better in the afternoon than in the morning. And there's been countless studies looking at this effect. I've listed just a few of them here, looking at swimming, tennis, soccer, flexibility, but big picture, the literature has found that people perform better later in the day than earlier in the day. And people have proposed that skeletal muscle is the limiting factor. And so one study that has looked at this is they looked at one rep max strength in 3 different exercises at 2 different times of day. So they looked at leg extension, leg curls, and the squat at 7 a.m. or at 5 p.m. And what they found is that the subjects performed better at 5 p.m. than they did in at the morning routine. And there's been countless studies that have come to the same conclusion, which is that exercise performance varies by time of day, and they assume it's because of intrinsic changes in skeletal muscle. And by intrinsic, I mean that the muscle is what's being regulated, and that muscle is the limiting factor. There's a huge issue with this, though, which is that no one has actually done direct muscle testing at multiple different times of day throughout the circadian cycle. Because, for example, in this study, there's much more at play than just the muscle. There's the role of neuromuscular factors, of body temperature, which changes by time of day. They came to the lab fasted. So they've been fasting for, several hours at that point where they were allowed to eat whatever they want for the rest of the day before the other time point. So there's a lot of third variables here. So to say that muscle is the rate limiter or the limiter of exercise performance and that it's regulated by time of day, that's a question that can't be answered specifically by a study like this. And so for our purposes, we define an intrinsic time of day change as one that is present in muscles of various myosin heavy chain compositions, because a lot of studies only look at one type of muscle. So there's no data as to whether the effect that's seen in that muscle will look the same in a different muscle with different fiber type. So that's our first criteria, that's present in different muscles of various myosin heavy chain compositions, and then it's also present during ex vivo contractile testing. So one of the big benefits of doing ex vivo testing is it controls for those things that I mentioned earlier. It controls for the neuromuscular factors because we're bypassing that. We are taking the muscle out of the mouse, which we used for our model, for example, and stimulating it with electrodes instead of a nerve. So you're bypassing that. You're controlling for muscle temperature, oxygen delivery, carbohydrate availability, and it really comes down to just the muscle is the limiting factor. And so our first study that we did had the main question of, does time of day regulate intrinsic contractile properties or contraction-stimulated glucose uptake in skeletal muscle? And based off of the literature in the field, that seems to overwhelmingly support that performance varies by time of day, that it's better in the afternoon than in the morning, and then it comes down to muscle. So going along with that, we hypothesized that time of day would regulate contractile function and contraction-stimulated glucose uptake. As I was kind of talking about earlier, there's some major gaps in knowledge in the field, which is essentially that intrinsic muscle function hasn't actually been measured at different time points of the circadian cycle. And there's a few different ways you can look at intrinsic muscle function. So you can look at force production, endurance, and we also looked at contraction-stimulated glucose uptake. And so this is a little schematic of our experimental design. So big picture, we had 3 primary measures. So basically looking at force production, endurance, and glucose uptake. And we did those measurements in 2 different muscles in mice. We chose the soleus and the extensor digitorum longus because they have very disparate fiber type compositions, where soleus is more slow twitch, and extensor digitorum longus is more fast twitch. So that kind of gives us 2 ends of the spectrum of fiber type composition. And we did all these measurements at 4 different times of day. And so you might notice I have things labeled ZT1, ZT7, ZT13, etc. ZT stands for Zeitgeber time, which in the field of chronobiology basically describes the light dark cycle of the vivarium. And it's kind of just a way to standardize the way people report time in experiments. So for a typical vivarium, there's a 12-hour on, 12-hour off light cycle, and lights turn on at ZT0 and they turn off at ZT12. And that's what our vivarium does. So that's what we used for our experiment. We chose 4 different time points, evenly spaced throughout the circadian cycle, with the idea that we'll be examining both the light phase and the dark phase, which encompass the 2 activity phases for mice. So mice are mostly sedentary and don't eat quite a lot of food when the lights are on because they're nocturnal. Most of their activity and most of their feeding behavior is at night when the lights are off. So we wanted to make sure we're getting both sides of those spectrum, but we also wanted to make sure that we were looking at multiple points within that spectrum. So for each phase, we have basically an early and a mid to late time point within each light dark phase. And we did this in male and female mice, and we did this after a standardized last meal in a fast or without a standardized last meal, but still a fast. Basically, for purposes of simplicity, I'm just going to go through the female data after a last meal, but big picture, we didn't see any difference between sex or between feeding status. So the findings that we have in that subgroup apply to all of these other groups. And so just to kind of zoom in at the experiment that was done at each time point. So we removed both EDL muscles, so EDL is short for extensor digitorum longus, and both soleus muscles from each mouse. The muscles from one leg would just be rested, whereas the muscles from the other leg would be taken to the ex vivo contraction apparatus. And they first would do a stress frequency curve, which looks at the contractile properties of the muscles. And then the muscles had a 10-minute rest to recover from the stress frequency test. And then they started an endurance test where they received repeated contractions over a 15-minute period. And you can watch the muscle lose its ability to produce stress over time. And for this first kind of period here, where we remove the muscles and we're letting them recover, and during the stress frequency testing, it was in what we call recovery KHB, which is basically just regular KHB buffer. However, during the endurance test, or for the last 15 minutes of the experiment for the rested muscles, they were incubated in what we have labeled INC KHB, which has radio labeled 2-deoxyglucose that allows us to measure glucose uptake during the contraction test. And so this should be a video here. Yeah. So this is just kind of a little view of what the muscle looks like as it's contracting in the chamber. So this is a EDL muscle. You can see the muscle origin tied to a rigid post here on the right, and the muscle insertion is tied to a force transducer on the left. It's flanked by 2 platinum electrodes, which produce a voltage at various frequencies and cause the muscle to contract in that buffer solution there. So I think it's pretty cool to just see the actual muscle contracting in the chamber. And so the first thing that we wanted to make sure that we looked at is that the circadian rhythm was actually present in the mice that we tested. And so the circadian rhythm is is regulated by an actual molecular clock that is in the main clock is in the brain, but basically every tissue in the body has a molecular clock in it that has oscillations of different proteins that basically go up and down on a 24-hour cycle. And that's what basically are the gears of the circadian clock. So we first wanted to make sure that the circadian rhythm was actually robustly entrained and preserved in the mice that we were testing. So we looked at multiple different muscles, so EDL, soleus, gastrocnemius, and we looked at a non-muscle tissue in liver. And we see that for this core clock protein BMAL1, you can kind of see what I was talking about. So we have ZT1, 7, 13, 19. As you go throughout the day, so the yellow is light, blue is dark, you start off with a high level of BMAL1 being expressed in these different tissues. It goes down throughout the day, and then during the night phase transitions back to being high again on a 24-hour cycle. And we didn't only just look at BMAL1, the core clock gene, we also looked at DBP, which is a clock output gene. It's a gene that's regulated just downstream of BMAL1. And you can see there's the characteristic inverse relationship between BMAL1 and DBP, because the circadian rhythm is composed of these transcriptional translational feedback loops where when one protein is being expressed, a lot of times it represses its own repressor, which basically results in them going up and down in opposite directions throughout the 24-hour cycle. And you can see that represented here in BMAL1 in DBP. And just to be sure, we measured another core clock gene, NR1D1. And again, we saw that throughout various muscles in different tissues, there was robust time of day regulation. So we were certain that the mice that we were testing had a robustly entrained and healthy circadian rhythm in these various different tissues. And so, as I mentioned, kind of in our little flow chart of the experiment, the first thing that we did was a stress frequency curve. So we stimulate the muscle at different frequencies, starting from a single twitch contraction, so 1 hertz frequency, just sending a single electrical impulse. And then we do increasing frequencies up until the muscle basically plateaus at its maximal stress production. So you can see as we increase in frequency, you see an increase in stress produced by the muscle. We see that demonstrated in both soleus and EDL, and there was no difference in the curves at any different time of day that we tested it. So maximal and submaximal force production are not regulated by time of day. We also looked at intrinsic muscle endurance, and this is the test that followed the stress frequency curve where we contracted the muscle every 15 seconds. And over time, you can see that the muscle slowly loses force until it plateaus. And so you can both kind of look at the curve and see that there's not a difference based off of time of day, or you could take the area under the curve, and that's just another way to basically show time of day doesn't affect the intrinsic endurance capacity of muscle. And we did this in both EDL and in soleus and saw the same effect. During the endurance protocol, we measured contraction-stimulated glucose uptake. So that's why we have those paired rested muscles included in the experiment, so we can compare basal glucose uptake to contracted muscle glucose uptake. And you can see that in the contracted muscles, there's a significant increase in glucose uptake. However, this increase was not variable by time of day. So these values here are what we call contraction-stimulated glucose uptake. So it's taking the subtracting the rested value from the contracted value. So these are basically the deltas between these different columns. In big picture and in EDL and soleus, we saw robust contraction-stimulated glucose uptake, though it was not regulated by time of day. So the major takeaway from this first study is that the contractile properties of skeletal muscle, the endurance properties, and the ability to do contraction-stimulated glucose uptake are not intrinsically regulated by time of day in muscle. So this suggests that the effects that people have described on athletic performance, for example, are likely not due to skeletal muscle itself. It's more likely due to some of those other third variables that we talked about earlier. So that's study number one. For study number two, we wanted to look at mitochondrial function in skeletal muscle and see if there is intrinsic circadian regulation. So based off of the results from study number one, we kind of pivoted our thinking. So no, we did pivot, but I said that we thought it would regulate. We thought it would not, that's a typo, regulate intrinsic mitochondrial function in skeletal muscle based off of the findings in the last experiment. And so our definition of intrinsic time of day mitochondrial regulation is that the change must be present in muscles of various myosin heavy chain compositions. So that's similar to study number one. But in this one, it must be present after a standardized last meal. Thousands of genes can change after you eat a meal or have a glucose load or something like that. So to normalize for that and the effect of fed fasted status on gene expression and mitochondrial function, for this study, we did include that standardized last meal before all of the time points. And so major gaps in knowledge are basically that no one's done it. No one's actually measured markers of mitochondrial biogenesis, dynamics, or metabolism at different times of day in post-absorptive muscle. So after this standardized meal and fast. And there's been no actual direct testing of mitochondrial function at different times of day. So this study design is pretty similar to study number one. We use the same exact time points, ZT1, 7, 13, and 19. And we measured markers of mitochondrial function at the gene level and at the protein level. We also did functional measurements such as maximal uncoupled respiration and SDH activity. And we did this in a wide variety of muscles. So we did them in soleus, EDL, gastrocnemius, tibialis anterior, and plantaris. And so some of the key measurements that we did is we used RT-qPCR to look at transcript abundance of various mitochondrial markers in EDL, soleus, and gastrocnemius. We did immunoblotting for electron transport chain complex abundances in EDL, soleus, and gastroc. And we did those direct functional measurements in TA and plantaris. And so the first thing that we did is we measured the transcript abundance of a panel of genes, including BMAL1 and DBP, which are, as I mentioned earlier, BMAL1 is a core clock gene and DBP is a clock output gene. So we'd expect to see significant circadian regulation of each of those with an inverse relationship between them. So those are basically our positive controls for this experiment. You can see that in EDL, in soleus, and in gastrocnemius, while BMAL1 in DBP had robust, significant time of day regulation of their expression, this whole panel of genes involved in biogenesis, carbohydrate metabolism, lipid metabolism, mitophagy, mitochondrial fusion, fission, basically all the things that people say are regulated by time of day, but haven't actually done the measurement. We did, and they don't change by time of day. We also looked at transcript abundance of electron transport chain complexes, and we did find some subtle muscle specific in EDL or gastroc, but we did see some differences in the soleus. So we saw some subtle yet significant circadian rhythmicity of complexes 1 and 4 in soleus. And this finding was really interesting. This is one of the first um, instances. I don't like to say positive or negative because that kind of makes me think good or bad. There's no good or bad data. The data is just data. This is like our first positive data that we saw. And going back, that made us want to look at basically the changes in expression levels between the different muscles. We noticed that those were actually different. So that's a finding that we also just have kind of added to the field and is a big strength of us looking at multiple muscles as opposed to one muscle. So, for example, you can see that basically expression of BMAL1, for example, let's just zoom in on the soleus, is basically fourfold that in the gastrocnemius or in EDL, which hadn't been described before, which is pretty interesting and suggests that fiber type may play a role in the amplitude of the circadian rhythm, which had not been reported before. And so for the electron transport chain complexes, we not only looked at transcript abundance, but also looked at actual protein abundance of the complexes. So here I have the same complexes that we measured using RT-qPCR, complexes 1, 2, 4, and 5. We also did using immunoblotting, and we saw no difference based off of time of day in EDL, soleus, or gastroc. So EDL and gastroc kind of go with what we saw on the qPCR data. In soleus, I'll just remind you, complexes 1 and 4 were significant in qPCR, but they weren't significant on the Western blot. So I just think if there's anything to take away from that, it's the importance of following up uh, gene expression level data or transcriptomics data with actual measures of protein abundance, because it's not always a direct comparison. Because in this case, it seems that the subtle variation that you see, I think it's about a 50% increase in amplitude throughout the circadian cycle, doesn't actually reflect in time of day changes at the protein level. We also looked at some direct measurements of mitochondrial function. So one thing that we did is we had some awesome collaborators help us do some measurements of oxygen consumption rate using the seahorse machine. And we looked at maximal uncoupled respiration for complexes 1, 4, and 5. And we saw no effect of time of day on maximum uncoupled respiration. We also looked at succinate dehydrogenase, which is complex 2 activity, using histology. And we saw no effect of time of day on SDH activity either. So the major takeaways from study number 2 are that intrinsic mitochondrial function is not regulated by time of day, be it at the gene, protein, or functional level. And the big picture takeaway is that time of day effects on exercise performance and metabolism are likely not due to intrinsic circadian regulation of mitochondria. So that's study number 2. In study number 3, so now we're kind of going to use our magnifying glass and look more mechanistically at how the molecular signaling of contraction-stimulated glucose uptake works. So we looked at these two proteins, P300 and CBP, and we hypothesized that they would be required for contraction-stimulated glucose uptake. And there's a couple of reasons why we focused on P300 and CBP. The first one is that P300 and CBP have previously been shown to be absolutely required for insulin stimulated glucose uptake. And so this is data out of our lab from a paper done by Vitor, the previous MD PhD student in the lab. And a lot of his research focused on the roles of P300 CBP in insulin stimulated glucose uptake. And you can see that in both Soleus and in EDL, there is a robust response to insulin in these white bars, which are just regular wild type muscle. However, in these black bars, which are from mice with double knockout of P300 and CBP, insulin stimulated glucose uptake is completely blocked. And you can see that just further simplified in this graph of insulin stimulated glucose uptake, which is that delta between basal and insulin. So P3 and CBP have previously been shown to be required for insulin stimulated glucose uptake. We thought about P300 and CBP in relation to contraction stimulated glucose uptake because muscle contractions can stimulate glucose uptake independently of insulin, but the way that the mechanism works, the final steps of the process of how glucose gets into the cell are the same. So we thought that there might be some intersection between these two signaling pathways. So as I mentioned, contraction-stimulated glucose uptake can happen independently of insulin signaling. So in this study, they looked at wild-type rats and rats with muscle-specific insulin receptor knockout. And they're looking at that delta here. So you can see a large increase in glucose uptake in response to insulin in the control mouse that's blocked in the mouse lacking the insulin receptor, unsurprisingly. However, both the wild-type mouse and the mouse lacking the insulin receptor have significant and robust glucose uptake in response to exercise. And both insulin and contraction result in glucose uptake into the muscle cell by GLUT4 translocation from an intracellular vesicle to the plasma membrane, where the transporter allows for the facilitated diffusion of glucose from the blood into the cell. And you can see that this GLUT4 translocation to the plasma membrane is preserved in both people without type 2 diabetes and people with type 2 diabetes. When both groups of people exercise, GLUT4 is translocated to the plasma membrane. So contraction-stimulated glucose uptake is preserved. And so we think that P300 and CBP are promising candidates for regulation of C-stim glucose uptake for a couple of reasons. One is that they have a strong cytosolic presence and directly acetylate cytosolic proteins. This is important because protein acetylation is a post-translational modification that can change protein function, just like phosphorylation can. And the cytosol is where GLUT4 trafficking happens. So it's important that these proteins be in the cytosol. They're also regulated by various kinases that can be activated by exercise, such as CAMK4 or AMP kinase or P38. And we wanted to look at both of these proteins, P300 and CBP, because they're paralog proteins, which means that they have very similar sequence homology. Overall, they have 61% of the same sequence. And in their acetyltransferase domain, they're 86% homologous. So in a way, they're fraternal twins of sorts. The idea or the leading theory is that this their duplication is a result of a duplication event that happened at some point that was evolutionarily conserved because it's good to have multiple copies of these genes, which suggests that they're very important for something related to survival. And so the current thinking in the field is that contraction stimulated glucose uptake is mediated by phosphorylation based signaling. And there's several different proteins that are phosphorylated in response to exercise that people hypothesize would be involved in contraction stimulated glucose uptake. The issue is that when you knock out these proteins, you either have a decrease in glucose uptake, usually on the order of 10 to 30%, or it has no effect at all. Another thing that people have looked at is the role of mechanical tension, so the actual force production by the muscle as a signal, but that's been pretty debunked because you can use people have done experiments where you inhibit actin and myosin cross bridge formation, and the muscles produce zero force, but they take up the same amount of glucose in response to the contraction stimulus as muscles that produce normal force. So mechanical tension is pretty conclusively a non-factor in contraction-stimulated glucose uptake. And people have done knockouts or used pharmacological inhibition of enzymatic activity of these different proteins. And whichever way you slice it, they either have a very little effect or no effect on contraction-stimulated glucose uptake. So it's still a really big question in the field as to what is the signal, or even is there a fundamental regulatory signal, or is it just highly redundant and compensatory because glucose uptake is very important for survival? So those are kind of questions that people have been posing. But based off of our data in insulin stimulated glucose uptake, showing that they're absolutely required fundamental regulators of insulin stimulated glucose uptake. And because of the intersection of the distal ends of the insulin and contraction stimulated glucose uptake pathways, we hypothesized that P300 and CBP would be fundamental regulators of contraction stimulated glucose uptake. By fundamental, we mean that P300 or CBP would be required for contraction stimulated glucose uptake. So if you knock them out, there's no glucose uptake, and that we would see this effect in both male and female mice and in muscles of various myosin heavy chain composition. And so to do this, we used our P300 and CBP mouse knockout model. And in this model, it's a inducible skeletal muscle specific knockout of either both alleles of P300 and both alleles of CBP, all but a single allele of CBP, and all but a single allele of P300. So these different models basically help us to look at the fundamental role of P300 and CBP with the double knockout, and get to see basically how do the muscles react when we rescue a single allele of P300 or CBP. And so for this study, we did similar methods to what we did when we measured contraction-stimulated glucose uptake in the Circadian studies. We dissected out bilateral EDL and soleus. The muscles from one leg would be rested, the others would be contracted, and either during the 15 minutes of contraction or the 15 minutes of resting, they would be incubated with a radio-labeled glucose tracer that we can use to measure glucose uptake. And what we saw is that contraction-stimulated glucose uptake was blocked in the IM-PC knockout mouse, so the P300 CBP knockout mice. So we saw robust glucose uptake in response to contraction in the wild type mouse, but this effect was blocked in the knockout mouse. You can see that when you do the delta between rest and contraction, there's robust glucose uptake in wild type, but the C-stim glucose uptake in the knockout was not statistically different from basically a normal distribution around zero. So it was completely blocked. We saw this in both EDL and in soleus. So we next wanted to look at the importance of a single allele of P300 and CBP. And we saw that bringing back just one allele of P300 or one allele of CBP was sufficient to restore glucose uptake to wild type. So we have the single allele of P300 up in this top row and the single allele of CBP in the bottom row. And as you can see, they look no different from their wild type litter mates that have all four alleles of P300 and CBP. So since P300 and CBP are acetyltransferase enzymes, it's important to distinguish their indirect effects from their direct effects. The indirect effects are because they acetylate lysines on histones, which can alter gene transcription and therefore protein abundance and function, which could lead to the phenotype that we're seeing. The direct role would mean directly acetylating a protein, changing its function that immediately results in a change in physiology and metabolism. And with the knockout models, we're not able to discriminate between the two. So we wanted to come up with an experiment to do that. And the way that we did it was with acute incubation of muscles with IP300W, which is a novel P300 CBP inhibitor of acetyltransferase activity. So we performed the same glucose uptake experiment in muscles from wild type mice where half were incubated with vehicle and half were incubated with this inhibitor starting 60 minutes before the contraction and then lasting during the contractions. And what we saw is basically identical to what we saw in the knockout models. So we saw that C-stim glucose uptake was blocked with IP300W in both EDL and in soleus. So you might be wondering, what about these other signaling proteins people have put forth in literature? Well, we measured those too, and I measured all of the proteins in the blots on the right, but I'll just use one for the sake of time. But we saw that big picture, they were unaffected by the drug in that the signaling was preserved. So for example, we see P38 MAP kinase activation. It's activated robustly in response to contractions, about a fourfold activation. In IP300W, we see a slight increase in basal P38 activation, but that's more likely a secondary result of the inability to take up glucose into the cell because P38 is very sensitive to oxidative stress. So it basically is providing some insight onto that aspect of things and suggesting that the muscle is experiencing some stress due to the inability to take up glucose, but the signaling is still there and glucose uptake is blocked. So it's unlikely that, and we see the same for basically all of these different signaling molecules that we tested, except for hexokinase, which is not a signaling molecule, but is the rate limiting step of glucose uptake into the muscle. And that was unaffected by P300W. But all these signaling molecules, the signaling was preserved. So the major takeaways from study number 3 is that P300 or CBP is required for contraction-stimulated glucose uptake, in that P300 and CBP likely regulate C-stim glucose uptake through their direct effects, not their indirect effects. So that's study number 3. And I just like to say, if you take away 3 things from this presentation, I hope you remember that. 1, the intrinsic contractile properties of skeletal muscle are not regulated by time of day. That contraction-stimulated glucose uptake and mitochondrial function are not regulated by time of day. And that P300 and CBP are required for contraction-stimulated glucose uptake. I have a lot of people I would like to thank for helping me do this work. I basically didn't know how to do anything except pipette. I'd never worked with mice. I'd done like 3 Westerns in my life when I joined the lab. I'd never done muscle mechanics. And I learned so much from a bunch of really awesome people, and they're all listed here on this screen. So I'd just like to thank everybody on this screen. And I'm happy to take any questions.
Nice job, Liam. Thank you for cutting it a little short too, in terms of, I know we started a bit late given the Zoom issues. So great job with pulling all your thesis stuff together. Are there any questions people would like to ask based on his presentation? Samir?
Really great job, Liam. This is it's very hard to tie up supposedly negative results, but you did a really nice job explaining the logic. So
Thank
well
you.
done. Couple of quick speculative types of questions. Not expecting you to have the data to answer these questions, but just in terms of what else you've read or observed in your findings. First, do you think baseline metabolic state plays a role in any of the circadian related outcomes? So take apart the fact that it's muscle or nervous system or vascular system or what have you. If someone is an elite athlete versus an average person versus someone with a baseline existing metabolic syndrome, would the would it just mute the regulatory effects, or would this be something that's um, shifted or enhanced, or I guess how would you think about that?
Yeah, that's a good question. I think on the kind of molecular regulatory level or molecular signaling level, I wouldn't, zooming in on muscle, specifically a muscle. Based off of our data and the data that I've seen in the literature, I'm not convinced that there would be a huge effect. But I think this kind of gets at one of the things that the field needs to be more specific on, and is kind of a conversation that we've started, is basically discriminating exercise from muscle function, because there's a lot of different components that go into exercise. And kind of what you're talking about, a sedentary person versus an active person versus an elite athlete, I would not be surprised if there were differences in athletic performance based off of time of day, because there are a lot of different um, studies showing that particularly training time can have an impact. So if they take one group, like there's a study where they took 2 group of people and they took morning and evening uh, measurements of different exercises, and then had them specifically train at either the morning or the evening for 6 weeks, and then redo it. And the adaptation to the training, they both adapted. They both got better at the exercise, but they got better at whatever time that they trained at. So I think it's not more of there's difference between an elite athlete's intrinsic muscle function versus a active person's muscle function on an intrinsic level. But I do think differences in training, and certainly differences in basically like central nervous system activation to do it could change between people. And I think that that's likely the difference maker between an athlete and someone who just, woke up at 6 in the morning and is trying to go to the gym and goes and feels like crap and feels weaker. I think athletes have a lot of training, both mentally and in their nutrition, basically to do what they need to do to prepare for an event if it's early in the morning. That can offset a lot of those drawbacks from early morning training that someone who's not an athlete or someone who loves training might
Cool.
experience. So that's
Thank
my
you.
that's my basically my guess.
That works. Thank you. And I have to hop off, so sorry to just rush off, but great job
Thank
again.
you.
So maybe I missed that point. Is like when you use the acetylation inhibitor, do you see the basal glucose uptake decrease or change it? I can't remember exactly.
For which part? I measured glucose uptake in the circadian glucose uptake or in the
Or the inhibitors, that's the P300 or CBP inhibitors.
yes. This one?
Yeah.
Yeah, it was blocked. There was no, the basal glucose uptake was the same as incubated with drug, for example, or this is the drug one.
Yeah. So how long is the treatment?
Treatment's for 1 hour.
Pre-treatment for 1 hour. Okay, interesting.
Yeah, it's for 60 minutes, with the idea being that that's not enough time for there to be appreciable changes in transcriptional regulation inside the cell. It's an acute inhibition of acetyltransferase, specifically on P300 and CBP.
So you think that during the contraction, somehow this acetyltransferase is actually activated?
Yes, and we do know that P300 and CBP can be activated by various kinases that in turn are activated by contraction. So there is a kind of a paper trail you could follow there to hypothesize that.
Severin?
Yeah, I was wondering if you ran like in your knockout model, if you ran in a chicken or the egg problem when you say that P300 CBP are essential for glucose uptake, like, and then you knock it out, you basically cut off glucose as a substrate, right? So that would affect the quality of the muscle contraction.
Yes, that's true. We did, for example, we did measure so we did measure contractile function, for example, in the IP300W experiment with the acute inhibition, and we saw no difference between vehicle and IP300W.
And do you know whether these mice are able to adapt to utilize different substrates for contraction?
We have not done that experiment. We've just focused on glucose uptake.
Thank you. It's very interesting.
Well, importantly, though, in the buffer, there isn't glucose in the buffer during the contraction. It's pyruvate, and so it's independent of the ability of the muscle to take up. In terms of there is glucose substrate, there is a pyruvate substrate for them to use in the buffer for the contraction portion. But as part of that, you're also measuring glucose uptake through of glucose itself. But the muscles can technically use other substrates. Any other questions? I know we're over time, so thanks everybody for your patience with the start of the talk, and great job, Liam. And good luck, obviously, in a couple of weeks, and we'll make sure everyone in the department knows when your talk is as well. So great, once again, great job, and thank you for everyone for attending. Thanks.
Awesome. Thank you, everybody. Appreciate it.